The aim of this experiment is to assess the efficacy of antibiotics against Pseudomonas aeruginosa biofilms under conditions representative of chronic infections in the cystic fibrosis lung. Using a defined artificial sputum medium, bacterial biofilms are grown in 24-well microtiter plates. The biofilms are then exposed to antibiotics. Next, antibiotic-treated cultures are disrupted using cellulase and incubated with a metabolic dye, risazorin, which is reduced from blue to pink by viable cells. By measuring the absorbance of the wells, the biofilm sessile cell minimum inhibitory concentration, or BSMIC, can be determined to quantify antibiotic susceptibility under near physiological conditions.
The main advantage of this technique over existing methods such as the determination of minimum inhibitory concentrations based on planktonic culture is that the bacteria are grown in a specialised media containing all the components of sputum, thus promoting biofilm formation. These biofilms are much more resistant to antibiotics.
To make the medium, first prepare 2 bottles containing 250 millilitres of sterile water. In the first bottle, gradually add the stirrer and then add 4 grams of fish sperm DNA to 250 millilitres of sterile water. Stir the solution at room temperature and allow the DNA to dissolve. To the second bottle, gradually add 5 grams of type 2 mucin from porcine stomach and stir. Once the DNA and mucin solutions are ready, dissolve the amino acids in 100 millilitres of